The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome. We will start today's lesson, Assaying Genome Diversity, by answering the following question. Why study genetic variation? Population and evolutionary genetics uses patterns of genetic differences between species, populations, and individuals to make scientific inferences. For example, human population genetics can help to explain the distribution and frequencies of different phenotypes and diseases in humans today. Information from genetic studies, placed in the context of information from other scientific disciplines, provides a synthetic view of evolution of diversity. Finally, asking clear, answerable questions based on verifiable evidence is an important aspect of studies of genomics. Genotyping is the process of determining differences in the genetic makeup of an individual by examining the individual's DNA sequence using biological assays and comparing it to another individual's sequence or a reference sequence. Genotyping reveals the alleles an individual has inherited from their parents. Early genotyping was PCR-based and used either presence versus absence of application or variable length markers such as RFLPs, microsatellites of variable nucleotide repeats. On PCR, RFLP is a simple low-throughput method. In this hypothetical example, a G versus A SNP creates or destroys a restriction site for restriction enzyme BAMHI. A second constitutive site within the same PCR amplicon provides a positive control for the completeness of restriction enzyme digestion. Another low-throughput method is RTQPCR. A real-time polymerase chain reaction, RT-PCR, is a laboratory technique of molecular biology based on the polymerase chain reaction of PCR. It monitors the amplification of targeted DNA molecule using PCR. In other words, in real time and not at its end, as in conventional PCR. The real-time PCR can be used quantitatively, or quantitative real-time PCR. Semi-quantitatively, in other words, above-below a certain amount of DNA molecules. Please do not confuse real-time PCR, denoted RT-PCR, with reverse transcription PCR, RT-PCR. A medium throughput method example is primer extension and MALDI. Matrix-assisted laser desorption ionization, or MALDI, examines changes in molecular weight of primers caused by base incorporation time taken for these ions to traverse a time of flight, or TOF. We're monitoring incorporation of a specific base complementary to the SNP by measuring the mass-charge ratio of the product by mass spectrometry. The method allows multiplexing of tenths of SNPs. Another medium throughput method that's often used is multiplex typing of microsatellites. In this method, PCR primers labeled with fluorescent dyes allow multiplexing and simultaneous detection of several loci, in this case 4, on a capillary electrophoresis platform. Primers amplifying loci with non-overlapping size ranges, here they're in blue, can be labeled with the same dye. A fourth dye is used for a size standard, not shown here, which is run in every lane. 5-dye systems are also available, which allow deeper multiplexing. A high throughput method is based on primer extension, An example is Infinium assay used in Illumina bead chips with assays up to 1.2 million SNPs. Following whole genome amplification, DNA fragments are allowed to anneal to an array of silica beads, each of which carries many copies of 50 nucleotide oligonucleotide whose 3' end lies adjacent to a particular SNP site. Primary extension incorporates the complementary modified D-deoxynucleotide, or ddNPP, which terminates synthesis, and also allows detection via fluorescence. Homozygous SNPs will produce single-color fluorescence, while heterozygous SNPs will produce equal ratios of 2 colors, allowing the genotype at each bead to be called. Another high-throughput approach is hybridization microarrays. An example of this is Affymetrix gene chips. In this method, oligonucleotides or oligos, usually 24 Mars or 24 base long, are directly synthesized onto a glass wafer using a combination of semiconductor-based polylithography and solid-phase chemical synthesis. Each array contains up to 900,000 different oligos, and each oligo is present in millions of copies. Since oligonucleotide probes are synthesized in known locations on the array, the hybridization patterns and signal intensities can be interpreted in terms of gene identity and relative expression levels by Affymetrix gene chip operating software. Now we're going to talk about sequencing to genotype. We can use whole genome shotgun sequencing to genotype. In this method, a random library of genomic fragments is constructed first, which contains platform-specific adapters on each end. The resulting fragments are sequenced directly at low coverage, allowing many individuals to be sequenced on the same chip or flow cell. Barcodes are used to separate between individuals or populations. Sequencing reads are aligned to the reference genomes, and then variation is identified computationally by comparing them to each other. In exome sequencing, we can gradually reduce an amount of information that can come from a single genome. So we will only sequence the exome, the only coding parts of the genome. To start, we need to capture the exome. In exome capture, a random library of genomic fragments, each containing platform-specific adapters at each end, is combined with a set of probes that define the human exome. If the hybridization is not by attached arrays, the probe, genomic library fragment hybrids, can also capture using magnetic beads and isolated from solution by amplification of a magnet or by solid phase capture. The naturing conditions are used to elute the captured genomic library fragment population from the hybrids and prepared for sequencing. Once more, sequencing reads are aligned to the reference genome, and variation is identified computationally by alignment. In case you do not have luxury of using exome capture, such as in species that have never been sequenced before, you can still use techniques like cDNA and transcriptome sequencing. Transcriptome sequencing provides an alternative of reducing the amount of sequence data for genotyping. In transcriptome sequencing, the RNA is first converted to cDNA, and the resulting cDNA are fragmented, and the library adapters are then ligated to the resultant fragments, followed by sequencing using next-generation methods. Since there is no reference, the transcriptomes need to be assembled de novo. Transcriptome sequencing can also be used by aligning to the reference genome, or exome, to identify differences in expression between individuals or tissues. Another way to approach a genome that has never been sequenced and reduce the amount of information is to use several reduced representation sequencing approaches. Reduced representation of genome is generally characterized by restriction digestion followed by direct sequencing at the ends of either a subset of size-selected restriction fragments or all of the restriction fragments in the genome at once. There are three general approaches, but all of them begin with a restriction enzyme digestion step. These are RRL, RAD-seq, and GBS. RRL, or Reduced Representation Library procedure. After digestion, the fragments from all samples are pooled and size selected to 300 to 700 base pairs. Then, fragments are ligated to standard sequencing adapters that are shown with gray squares and then sequenced. In this simple case, only the ends of fragments will be sequenced, but the protocol can be modified to sequence entire fragments. Either fragment ends, shown here with thick lines, or the entire fragments, shown here with thin lines, between 300 and 700 base pairs will be sequenced. Because the site at 1,300 bases is not present in sample 2, the long fragment between 700 and 2,000 base pairs is filtered by size select. In RAD-seq, or restriction site associated DNA sequencing, after the digestion step, fragments are ligated to P1 barcoded adapters. These will separate between samples, which are either individuals or population, here shown yellow for sample 1 and purple for sample 2. Then they are pooled, randomly sheared, and size selected to 300 to 700 base pairs. They are ligated to P2 adapters with divergent ends. So you can see here Y-shaped gray markers, and then they are ligated to the fragments with and without P1 adapters. PCR amplifies the RAD text selectively. Only the fragments containing the P1 adapter, unique identifier, a restriction site, and a P2 adapter will be amplified. After all this, sequenced are aligned and SNPs are identified or called through bioinformatic tools. Finally, genotyping by sequencing, or GBS. This method uses restriction enzymes coupled with DNA barcoded adapters to reduce complexity. GBS can simultaneously perform SNP discovery and genotyping with or without reference genome sequences. Barcoded adapters, here shown in yellow, and common adapters, here shown in gray, are ligated to digested fragments, producing fragments with barcode plus common, barcode plus barcode, and common plus common adapter combinations. Samples then are pooled and amplified on the Illumina Genome Analyzer flow cell. Only short samples featuring barcode plus common adapter combination is amplified for sequencing. Sequenced markers are aligned to the original reference genome, and SNPs are identified in the aligned fragments. At the end of this lecture, I provide you with some review questions. What is genotyping? What is low throughput and high throughput methods? What is the difference between RT-PCR and RT-PCR? What is the advantage of exome sequencing? What methods of exome sequencing would you use if there is no known genome for the particular species? Please give examples of reduced representation sequencing and discuss differences between them. This concludes today's lesson. Next, we will be studying the topics of pattern of genome variation mutations.